The team of University of Washington researchers working to find and characterize rotten sea ice traveled to the ice off Barrow, Alaska 3 times in the spring and summer of 2015.
What I was looking for in May was ice that had not yet been touched by the sun. So if it's flat and it's level and it's under snow cover, it hasn't really seen the light of day yet.
In June, they found melting ice.
And of course in June, you look off the beach and the first thing you see is this contrast between areas that are ponded and areas that are bare. And you immediately say that ice is different. And so the visual impression of it tells you a lot about the nature of the ice and the structure of it.
And in July, they found truly rotten ice.
In July, this ice was different. It had suffered considerably, and the surface of it was very rumpled, and the texture of it was very coarse. It was kind of riddled with drainage holes.
We really did find some very flat, uniform ice flows that were clear, and they were rotten through the whole length of the core. So that was exciting to see.
I had no idea what to expect because this was my first time on really on sea ice. And everybody previously that we had talked to had said, you're never going to be able to core this ice. It's going to fall apart, it's going to be all slush. We cored it, and it was intact ice with big holy bits that was breaking apart as we were standing on it. And as I was pumping the sample, this crack just goes and connects all the holes.
Yeah, the ice we found in July was more rotten than my wildest dreams.
It's so different just thinking about it versus actually having it in your hands and seeing it. And I, just like others in my team, was just amazed on how big those channels were.
Finding and working with rotten ice was difficult in July. Still, the team was able to run field experiments and prepare samples on the ice flows and in the Barrow lab, much as they had done in May and June.
Shelley started drilling the cores, and we drilled upwards of 22 cores each time we went out. We were collecting cores for doing melts to look at chemical and biological parameters. We drilled cores for optical measurements. We cut them up in little pieces to measure density and temperature. Monica was doing reads of photosynthetically active radiation to try to understand what the light environment was for the organisms in the ice.
Part of this project is to have this microscope set up in the cold room and look at these thin sections and look inside the ice to see the ice structure, but also the organisms within. In Barrow, we actually prepared the samples already. So these were ice samples that were melted in artificial seawater solution. So the osmotic shock of the organisms within is reduced.
It melts really slow. The bugs, we're assuming, are happier because they're not freshened. And then we can get accurate numbers when we do our processing for organisms.
Some samples are filtered to determine the amount of polymers in these samples.
We took optical cores while we were in the field, and we put those cores in sleeves and hauled them back to the lab. And I was able to work with them in the lab and cut them down into just 10-centimeter sections and look at the optical properties of those sections. People have been writing in their papers for quite a while now that in general the ice gets more transparent as the spring transitions into summer. And we know that that is certainly true for the ice cover as a whole, but is that really true about the ice? My hunch was that it wasn't necessarily true, that the ice should actually be less transparent as the season progresses. This ice is getting less and less transparent as it's warming, and the inclusions in it get larger and they scatter more light.
So now we are here, we've got the filters that need to be sent through the machines, need to be analyzed with the microscope or flow cytometer, or DNA needs to be extracted for further community composition analysis. And we also have course for the microscopy again. So we have the microscope back here set up in the cold room so we can make thin sections and develop more of these staining procedures to really get some good images of these organisms in the ice.
The cells and the gels, I will count with flow cytometry. A flow cytometer is an instrument that can measure and count cells or particles in flow. So you can see different populations of algae or bacteria, polymers, and you can see in time how these populations change.
We have beautiful profiles of temperature and how that changes with time, and same with salinity and the density of the cores. So the density of the May and June cores were very similar, but the July cores are a lot less dense, and that's because there's holes in it.
Right now we're in the business of trying to figure out what questions to really start asking about this new ice type, this rotten ice. And the first step in asking good questions is to have a good characterization.
Characterization of the physical, biological, and chemical nature of rotting first-year sea ice continues. Constructing a picture of how the ice and microbial communities changed during the summer may ultimately tell a story of how Arctic sea ice survives to the end of the melt season.
Science at work for you. This is APL, the Applied Physics Laboratory at the University of Washington in Seattle.